Hi, everybody. Thank you for having us here today. And we're pleased to be able to tell you a little bit about a project that we've been working on related to development of selection tools for male fertility in beef bulls. Before I move on, I want to recognize the contributions of Jenny Borman and Bob Weber, who've really been instrumental in this project. I'll also talk a little bit later on about some other folks that have been instrumental in this project as well. So to start with, I just kind of wanted to go through some of the things that we know about male fertility based on the scientific literature. Um, what we see in the literature is there's not a ton of estimates of genetic correlations between male and female fertility, but the ones that exist are a little bit older, but they do look like those genetic correlations are fairly low. So what that means is that when we take into account all of the great strides we've made in improvements in female fertility in the beef industry, that's probably not very likely to translate to male fertility. So we're going to need some separate metrics and separate tools to really be able to tackle those differently to make progress in male fertility. We know that characteristics of fertile males, which in this instance I'm classifying as things related to motility, ejaculate volume, abnormalities, etc., are lowly to moderately heritable in both beef and dairy bulls, although there are some differences in the heritability estimates between those two different groups. I think sort of intrinsically, we all probably recognize that the profitability of beef cattle operations for many, many people is directly related to the number of saleable calves that are produced. Um, so really that's a direct result of cows that conceive in the breeding season. And as a bonus, if you will, if we can get those cows to conceive earlier, we probably are going to have calves with larger weaning weights and thus probably have greater value in those particular calves, particularly if we're selling at week. We also know that favorable and unfavorable genetic correlations exist between production traits and semen quality traits. However, a lot of those estimates are from dairy populations, not necessarily beef populations. So I think we have a little bit more we can learn on that front. So if we think about why we might be interested in male fertility, if you look at the literature, there are some older estimates that show a pretty high rate, in my opinion, of BSC failures. And we know that probably no matter how much we select on a trait, we're not going to have zero losses due to infertility. But I think we could dramatically reduce the number of bulls that fail a BSC if we had dedicated male fertility selection tools that we utilized within the beef industry. And I think that would improve producer profitability. We also know that male fertility can be important, particularly a BSC, because a lot of insurance related to loss of use or bull mortality requires bulls to pass a BSC in order to qualify for those policies. And I think from a practical standpoint, if we have low genetic correlations between male fertility and female fertility, we've addressed the female side of the equation in many breeds and populations with some great selection tools. Why only address half of the equation, right? Let's develop those tools in male as well. So I'm going to argue that semen quality attributes are economically relevant for a variety of different reasons. The first of those being that we know that these individual semen quality metrics determine whether we can freeze the semen of a bull for artificial insemination. And so this is a big deal for these stock breeders that may be marketing bulls in that manner or marketing germplasm in that manner. And sort of within that, it's important to that particular seed stock breeder. But provided that that bull can sort of meet that threshold, at least, those genetics also trickle down to the rest of the beef industry. So sort of decisions or ability to meet those thresholds there really impact genetics within the rest of the industry. We also know that bull sellers cannot or maybe should not be selling bulls that can't pass a breeding soundness examination. And even if we have bulls that ultimately pass, even deferrals can be expensive, right? We may have to change our merchandising plan. We may have bulls that don't pass that BSC before a production sale deadline, right? And so they have to be marketed as 18-month-olds or later on in their lifetime, past yearling. And we also know that if we have bulls that fail to pass the BSC at all, we can take a big hit on those, right? Because by the time they're sexually mature and can undergo the BSC, we've already invested a lot of time and money into those animals. So if they ultimately can't pass it at all, those bulls can be castrated and sold, but we're going to take a big discount on those beyond the time and money that we've already invested in them as well. Both bull buyers and bull sellers need to get cows bred, right? We're selling animals. If we can't produce animals, we have nothing to sell. And I would argue that the herd sire, particularly when we're talking about natural service matings, really impacts fertility or the fertility outcomes of a herd more than any other individual or group of animals within the production system. And I think that makes sense, right? Because if we have a cow that has a fertility issue, we're missing a calf. If we have a bull that has a fertility issue that we don't catch prior to turning that natural service sire out, we've got a whole group of open cows. And while both of those things are important, we have a dramatically larger economic impact from the bull than we do from the cow. I think logically these semen quality attributes affect conception of a cow, but which of those traits are most important? And I think we have some things we can learn there. So if you're interested in any of these topics in general, one of our graduate students a couple of years ago put together a nice review on the subject in translational animal science. If you have interest, thought I'd plug that really quickly. I'd encourage you to check that out. So when we think about developing these tools, I think male fertility is kind of an interesting case because in many cases, when we're talking about development of, genetic evaluation for a novel trait in the industry, we've got to go out and collect a bunch of data. But the cool thing about male fertility is that the data actually already exists within the industry. So we probably have fewer, I hesitate to say no additional costs, but fewer additional costs for producers to be able to actually report that data, right? Because a lot of producers are already doing BSCs. What we really need is a pipeline to be able to submit that data to our genetic evaluation providers. We also have a situation where semen quality traits are routinely measured and evaluated within the industry. Now, there are a couple different ways we typically do that. First would be by bull studs in the process of collecting semen for AI. Now, the advantage and nice thing about those data is that they're typically much more standardized metrics, relatively consistent across different studs. But the disadvantage is that we're talking about a much more limited number of animals because it's going to be just bulls that we're trying to freeze semen for AI. And those data are often proprietary, right? They're not freely available within the industry. Now, our second pool of potential data here is collected by producers and their veterinarians when bulls undergo breeding soundness examination. Now, the disadvantage of this data is that it's much more variable in consistency because you have a lot of different people doing it in a lot of different places, and it may not all be recorded exactly the same, even though there are guidelines for doing that. But the nice thing about this data is that it's not restricted to AI bulls. It's really wide open within the industry, pretty widely available, and fewer issues around the proprietary nature of that data. So if we take a look at some of these specific metrics, what we see is that for those semen production traits, and by that I mean things like volume, concentration, and number of spermatozoa, are all going to be kind of lowly to moderately heritable in the industry. One thing that we do see in the literature is that oftentimes you'll see higher estimates, particularly for volume, in dairy cattle than we see in beef cattle. If we take a look at motility-related traits, what we see is that you typically have things like post-thaw motility and 3-hour post-thaw motility, more lowly heritable. And then things like initial motility and gross motility scores, kind of lowly to even moderately heritable for some of those types of traits. For morphology-related traits, what we tend to see is that these primary and secondary abnormalities tend to be kind of lowly to moderately. Head abnormalities a little bit higher in heritability, and tail abnormalities a little bit lower in heritability. And then total abnormalities a kind of range from lowly to moderately. Now, if we're going to develop male fertility selection tools, we've kind of developed some objectives surrounding this goal. So the first thing that we want to do is really try and understand the impact of specific semen quality and production metrics on conception in beef cows. So for that, we have a big group of beef on dairy and some beef on beef breeding records. We are really in the market for some additional records as well, where we can tie the semen collection metrics for that specific batch to those specific pregnancy outcomes. But we do have quite a bit of the other data. And one option for utilizing that is to develop a prototype kind of sire conception rate EPD. But I think really our ultimate goal for this particular pool of data is to better understand the component semen characteristics that influence conception in beef cows, and to be able to sort of relate that information back to some of the other tools that we want to develop in this project. Secondly, we really want to develop kind of a framework for a genetic prediction for male fertility in beef bulls. And two kind of pools of data that are going into that are some data that we have from a couple of different AI studs, including their semen quality parameters, and then information from breeding soundness exams on natural service sires. So with that, we hope to pull the knowledge that we've gained in that first bullet point with the metrics and the data from the second, with a particular emphasis on the data that's widely available within the industry, right, to develop some semen quality attribute EPD. And ultimately, what we may end up needing to do is to think about how to combine these different metrics within some sort of male fertility index. But at least initially, we're going to focus on those component traits. Now, in the context of providing this, one of the interesting things when you have genomic data that you can do is actually simultaneously identify and sort of validate either QTL, or which are regions of the genome that influence a particular trait, or the specific genetic variants within that region that we think are predictive for these component traits of male fertility. So in addition to genotype data, one of the things that we'll do in this project is we'll actually do some sequencing of some animals and imputation to full genome sequence variants wherever it's possible. And our goal here is to be able to not only identify those important genomic regions and candidate variants, but also to potentially identify some variants that aren't on the genotyping products that we currently use within the industry, so that those variants could go on chips that are built in the future, or possibly targeted capture when we're doing low pass sequencing. And finally, I think it's really important, if we're thinking about selecting for a new trait, that we consider what ties that trait has and what genetic correlations or particularly antagonisms that may exist between that trait and other traits that are economically important to us, right? Because we know we don't want to always select for one particular thing. We need to balance selection across a suite of traits. So if we are to add male fertility to some of our selection goals, what influence does that have on the other traits that are important to us? So what have we gotten done on this so far? We've published a few papers that look at some of these semen attributes. So I'll take you through and walk you through a few of the results on those particular papers, and we'll talk about some things that we have in progress as well. So we did begin with the Angus data in our first set of analyses. So the first group of data that we'll look at here had about 1,800 bulls. In reality, that number was a little bit lower because we did pull out bulls that were sort of custom collected that we didn't have sort of environmental data for. So those were removed. This data spanned 2004 to 2018. And ultimately, we ended up with about 44,000 individual collection records. And some of those traits had fewer records to them because not every batch of not every straw of semen that was frozen made it to some of these later collection metrics. So if we take a look here, we've got kind of a group of sort of semen production traits, if you will, that focus on the specific collection before it is frozen. So we've got volume and concentration and number of sperm. And then we also have a motility related trait in there. So initial motility would be as soon as the sample is collected prior to freezing. And then we have a group of sort of post-thaw metrics here. So post-thaw motility, and then some samples, a much smaller number, but some samples had a 3-hour post-thaw motility. So that sample sat for quite a bit longer, and then motility was measured again in that sample. And then some abnormality information. So percent normal, and then percentage of primary and secondary abnormalities. So for our particular genetic evaluation in these data, we tested a lot of different effects to go in the model. And the ones that ended up being the most significant that we included at the end were the collection location, which we had 2 different locations of collection, the day within the year that the collection occurred, and both of those were fit as sort of categorical variables. For covariates, we had the age of the bull at collection that ranged between just under a year old to some bulls of pretty advanced age. We had the number of days since the previous collection, which ranged anywhere between the bull being collected the day before and the bull being collected several years later. The average number here was 8 days between collections. And the cumulative cattle comfort index. So this range here reflects, this metric here reflects the cattle comfort index that was calculated for each day within the spermatogenesis cycle. And then we summed that all up with the idea to be able to account for the environment that each of those bulls experienced during the time when those sperm cells were being generated within the body. So to kind of account for the environmental conditions within that entire time period. We did test shorter time periods. We tested specific days, and the cumulative was the one that ended up being the most significant. So that's what we ended up with in the final. We did fit a genomic BLUP analysis, which was a single step. So we had both pedigree and genotype data in it within BLUP F90. We had a permanent environment effect. We had repeated records and repeated collections on these sires. And we tried to fit as many traits within a single model as possible. Most of the traits ended up being fit 2 at a time in bivariate models. One, we were actually able to get 3 of them to converge within a single trivariate model. So we'll look at that here in the next slide. So this first one highlighted in green, where we had volume, concentration, and number of sperm, was the suite of traits that we were able to fit in a trivariate model. So all of those were fit together. And you can see the heritabilities on those highlighted in the green box were fairly low, between 0.08 and 0.11. Now, the next set of models that we fit were all of those 3 motility traits. So the initial, when that sample was first collected, and then the post-thaw and 3-hour post-thaw. That would not converge as a trivariate model, possibly because we had a lot less data for that 3-hour post-thaw motility. So instead, we fit all of those traits in sort of pairwise comparison. So what you'll see there is that those were kind of on the upper end of lowly heritable, so a little bit higher than the production traits as a general rule. And then finally, we had the abnormalities fit. We tried to fit those in a single trivariate model. That did not work, so we ended up fitting them in pairwise bivariate model, 2 at a time. And the primary abnormalities, we would say, would be either very, very lowly heritable, or because of that standard error, 0 heritability in this particular population. But the percent normal was a little bit higher. And actually, one of the higher heritabilities that we got was the percentage of secondary abnormalities in this particular data set. The repeatabilities kind of range from 0.3 to about 0.6. So if we take a look at the genetic and phenotypic correlations between these, I'm going to focus on the genetic today for time purposes. But what you can see is that the phenotypic correlations are kind of in that lower left-hand triangular portion. I'm going to focus on the upper right-hand portion here and just highlight that we did have high genetic correlations between the number of sperm and volume and concentration, which I think is very, very logical. That makes sense from a phenotypic standpoint. Those correlations hold when we look at genetic correlations as well. So typically, as if we were to select for an individual with higher volume, we would typically get a larger number of sperm and a higher concentration as well. One interesting one here was between initial motility and post-thaw motility, very highly genetically correlated at over 0.9. So one of the things that I often think about and sometimes get asked about relative to male fertility is what about scrotal circumference? We have scrotal circumference EPDs, and in a lot of breeds, we've had them for a long time. So can't we just use that? So if you take a look in the literature, what you see in some older work is that serving capacity of a sire is not necessarily able to predict the fertility of yearling bulls as measured by pregnancy rate about 6 weeks into the breeding season. What we see as related to scrotal circumference most often in the literature is a relationship between scrotal circumference and younger age at puberty in heifers. Some studies have confirmed that. Others have found that that relationship maybe not as strong as in some of those initial papers. What I would argue, and this is my opinion, is that we don't really have a ton of compelling evidence that selection on scrotal circumference EPDs will dramatically impact male fertility. Okay. And part of the reason I say that is if you take a look at the genetic trends in a breed like Angus, what you see is that the genetic trend for scrotal circumference has been increasing as far back as like the mid 1980s, right? With an acceleration in that maybe around the mid 1990s or 2000. But if we take a look at the genetic trend for heifer pregnancy during that same time period, we see that it's pretty static for a lot of that time and actually takes a little bit of a dip in the late 90s. And then once that heifer pregnancy EPD was introduced, we see dramatic increases in heifer pregnancy genetic trend. So those two things don't necessarily mirror each other because those increases in the scrotal circumference genetic trend predate those increases in heifer pregnancy by about 25 years. So that's part of the reason that I say I don't think scrotal circumference it's certainly important in terms of making sure that bulls meet those scrotal circumference thresholds for sale. I don't know, based on what we've seen, that we're really getting at specific component traits when we're selecting on scrotal circumference. So that's one of the things we wanted to look at in this particular analysis that we just did. So what we did was we looked at the scrotal circumference EPDs produced by the American Angus Association and correlated those with our semen attribute breeding values that we produced in this particular study. And what we see is that there are really no strong correlations anywhere within this. The strongest would be between scrotal circumference and concentration and initial motility, but those are still not super high. Now, I will do a caveat here that this analysis is not as good as sort of fitting all those traits together in a multi-trait analysis. And that's something that we want to do in the future. But just sort of for a quick and fast version of that, we don't see any huge relationships here, which I think is really interesting and potentially important. Now, if we look at the results from some of the genomic work that we did, we were able to identify anywhere between 1 and 7 genomic regions that were significantly associated with these different semen attribute traits. I'm not going to bore you with this entire table, but I did just want to show you kind of where those shook out. So larger numbers for number of sperm, post-thaw motility, initial motility, percentage of normal sperm, spermatozoa and primary abnormalities, lower numbers for concentration, 3-hour post-thaw motility, and secondary abnormalities. Now, if we look at these from a more functional standpoint, what I've got in this table on the far left is the trait in our study that we identified that genomic region for. And then the SNP that was sort of representing that region is in the next column. But if you go all the way over to the right where it says QTL trait, that shows what trait, where there was already an association or a QTL within the literature for that particular trait. So for example, on this very first line, we found a SNP that was significantly associated with volume, and that genomic region overlaps one that had been identified in the literature as associated with conception rate. So if you take a look down this column, you see that we have a lot of SNPs that we identified, or genomic regions that we identified in our analysis that were already associated with something related to fertility within the literature. So things like non-return rate, conception rate, in some cases, scrotal circumference, daughter pregnancy rate, sperm average path velocity, a variety of different traits. A lot of those were dairy studies. But we are seeing some relationship between the regions that we identified and then other studies that have identified or looked for fertility related QTL within the genome. Now, if you take a look at the results, this is a Manhattan plot, and it's a little bit messy because we've got 3 traits that we fit in that trivariate model here. And what you see is that the light blue spots are volume, the yellow are concentration, and the kind of lavender purple ones are number of spermatozoa. And any of them that are above that line are above the significance threshold that we set for this particular study. So you see those SNPs from the table shown here within this graph. It's the same sort of setup in this particular study, only we had a couple of different analyses here for each trait. So you notice a post-thaw motility 1 and post-thaw motility 2, and that's because we couldn't get everything to converge within a single model, so we had pairs of bivariate analyses. But the same idea holds true here. These spots that you see above the line are the significant SNPs from those tables before. And then finally, if you see the exact same setup in this final graph for number of spermatozoa, or excuse me, normal spermatozoa, primary abnormalities, and secondary abnormalities. And so if a SNP was above that significance threshold in any of those analyses, we went ahead and looked at those to identify whether there were QTL that we could validate within the literature from those tables that we looked at before. Now, a couple of these we thought were extremely interesting. So this is the RS number associated with them, but the traits that we found significant associations with in these couple here that we thought were particularly interesting were motility traits, so initial motility and post-thaw motility, and then one for primary abnormalities. So that first one, if we take a look, there was the closest gene to that particular SNP was HERC2. And HERC2 has been associated with reduced spermatozoa motility in some other studies. And this particular SNP was within this gene. So we thought that one was a particularly interesting one to note. It is also downstream of OCA2. And OCA2 is a gene that contributes to spermatid development. And so those are both kind of within that QTL region for that particular SNP that was associated with both initial and post-thaw motility. Now, the second SNP that we found some interesting genes associated with was this one in the last line in the table. So it was significantly associated in our study with primary abnormality and was within this LOC gene without kind of a nice name like HERC2 and OCA2. But this particular gene has been associated with sperm motility, and the SNP that we identified as associated with it was downstream of that particular gene. So I think some really interesting candidates that could maybe be explored further in the future. If you have an interest in checking out either of these papers. This particular paper, if you scan that QR code, will get you to the paper that looked at the genetic parameter estimates, so the heritabilities and genetic correlations. The second one here gets you to the study that looked at some of the genomic data, so looking at those SNP associations. So before, as I kind of finish up the presentation here today, I want to tell you a little bit about some things that we're going to be doing as we move into the future. The first is utilizing some of the breeding data that we have to try and understand which component traits have the greatest influence on conception. So we do have a lot of beef on dairy data. We have a little bit of beef on beef data. So to give you an idea of kind of what that looks like, we have about 13 million dairy breeding records. If we filter it, it's pretty messy data. So if we filter it for a lot of missing information that's important for our analysis, we end up with about 4.5 million records. This includes around 1.2 million different cows and about 19,000 different sires. A lot of these are California dairies, but there is a variety of locations in the data. So we're looking at now how to sort of best use these records to achieve what we need to achieve here. So some of the future work that we're going to do with this is continue cleaning the data and test some criterion relating to the number of matings that we need to have per sire and all of those sorts of data quality issues. We're exploring whether we may need to request some more records. We're also looking at the exact same thing in some really high quality beef on beef records that we were able to get access to. If we need more data in either one of these areas, are there potential association partnerships or other data sources that we can explore? And then one of the first things we'll do with this is kind of explore what phenotypic effects are important for conception, with the dam as the animal of interest, and then doing that same exact analysis with the sire as the animal of interest, because we do need to sort of tackle the female side and the male side a little bit differently. So one of the other things that we're going to do is to build multi-trait genetic prediction models utilizing some of that BSC data that we have. So we have data, a bunch of data in Angus, but we also have a lot of data in some other breeds as well. So if we take a look at just the Angus data, we have about 5,000, a little over 5,000 BSC records from 13 different herds and 6 different technicians measuring those parameters. So if we take a look, we see proximal droplets and abnormal heads in the largest number, noted in the largest number of those BSC records. Detached heads and distal droplets in a much lower proportion of those records. DMR is also one that we see pretty frequently. But overall, 98.8% of these bulls were able to produce semen, and about 85% of them passed the exam in this particular case. So we do have some that didn't pass. So if we take a look at the summaries of these data, we have, again, those same about 5,300 records. About 2,900 of them have motility scored as a number, sort of like what you would see from a bull stud. So that is sort of detailed in this top table, along with the primary and secondary abnormalities. And then some samples only have percent normal and percent abnormal. So that's why you see a smaller number of primary and secondary abnormalities sort of broken out there. Now, about 2,200 samples, we have motility scores, but we don't have motility percentages. So for those, we have about 70% of the data falling into good or very good, about 9% in fair, and 10% in poor in that particular group of data. So in the future, we want to look at sort of developing that male fertility EPD and genetic evaluation framework using all of the data that we have available to us and focusing on which traits are the most critical and where we have data that can be sourced for the industry. Hopefully by the end of this, we will have the answer to whether we may need to be considering this as an index of a couple of different fertility traits. We'll also be sequencing some bulls that are influential within our data and our pedigrees. and then doing imputation and some additional genomic work. So before I sign off here and open any questions, I already mentioned Jenny and Bob, but I also want to mention Dr. Grieger. He's been really instrumental and is our go-to on asking questions about BSC data and some of the reproduction side of this particular project. And also JP Martin at the University of Wisconsin. He's been really instrumental in sort of understanding that beef on dairy conception data and working with us on some of those analyses. I would also be remiss if I didn't mention some of the students that were working on this. So the papers that you looked at were all written by Madison Butler. She was a fantastic graduate student that finished up a couple of years ago. Michael Simler is working with us to finish up his master's right now. And we have a variety of students that are working on the project right now. Danny is working with a lot of BSC data, Will with the breeding data. Elizabeth has worked with some of the BSC data in the past. And then Matt, Chantal, and Karis are just starting, but we'll be working on some different aspects of this project as well. And last but certainly not least, I want to thank USDA for funding this particular project and kind of moving us forward there. I'll take any questions if we can do that over Zoom. I'll leave that up to the organizers, but I will also put in a little plug. If you or someone you know has BSC records that you'd like to contribute to this particular project, I would be thrilled to have you email me at megrolf@ksu.edu. We'd love to have you as a part of this project if you have any interest. So with that, I will wrap up, and if we can zoom in questions, I'll be happy to answer those. Thank you very much for the invitation to tell you about this project.